Hey there, I'm Fritz, a PhD candidate from Hans-Peter Pfister's Visual Computing Group at Harvard University, where I work on visual exploration systems for genomic data. In this talk, I'm going to present Peaks, a tool for interactive visual pattern search in epigenomic data that is using unsupervised deep representation learning. This work emerged from a collaboration with Novartis Institutes for Biomedical Research and Harvard Medical School. When browsing one-dimensional epigenomic tracks, we often come across interesting patterns, like the one I'm showing here on the right, which can indicate the presence of a regulatory element. In many of these cases, we would like to find other genomic loci that express similar patterns to develop or test hypotheses or simply annotate datasets. While there are a lot of techniques for finding low-level patterns like peak detectors or finding specific features, general exploration of more complex patterns is non-trivial and not well supported in current systems. For this project, we focus on DNA-seq, a measure for DNA accessibility, and histone-marked ChIP-seq, which measures modifications of histone proteins, as together these two techniques can be used to identify regulatory elements like enhancers or active promoter regions. In general, searching for patterns in sequential data is challenging when the search space is large, the patterns are complex, or the query is difficult to formalize, which is all true in this scenario. To overcome these challenges, we need an effective feature representation that captures important characteristics of the data for similarity comparison. Such features could, for example, be the number, height, shape, or position of peaks, and are typically defined up front. While this is useful for finding specific patterns, it requires a deep understanding of the data and often does not generalize well to various different search queries. To improve upon existing solutions, we have developed Peaks. Peaks is a modular system consisting of two components: a deep learning-based data encoding strategy and a visual user interface for actively training a classifier to find other genomic loci that match the user's notion of similarity given a specific query. While there are many different ways to extract features from epigenomic data, we were wondering if we could leverage the remarkable performance of convolutional neural networks to learn a feature representation of the genomic data in an unsupervised fashion that is useful for pattern search. To this end, we've developed a convolutional autoencoder model that encodes a genomic region into a compressed feature representation. To provide a generic approach for visual pattern search, we built Peaks around an interactively trained binary classifier that operates in this learned feature space and tries to identify the relative dimensions for a specific query. The training data here for the classifier is provided interactively by the user in form of binary relevance feedback. Finally, since the search space is large, Peaks employs an active learning sampling strategy that selects regions that are assumed to be most beneficial for training the classifier. Now let's take a closer look at the encoding technique. We start by normalizing the signal and dividing the data into overlapping windows that are aggregated into vectors of size 120. We then pass each vector into our convolutional autoencoder model, which you can think of as an identity function whose objective is to reconstruct the input data. By forcing the autoencoder model to feed the data through a lower dimensional layer, the neural network has to learn a compressed latent representation, which we treat as the features of a genomic region. Now let's take a look at the user interface to see how all these aspects play together. At the top is the query view, which shows the genomic region we're searching for. This view is pan and zoomable and can show the autoencoder's reconstruction for debugging. The list view below shows several different genomic regions at once for comparison and labeling. The view features several tabs to access new samples, investigate the results, or browse already labeled regions. On the right side, Peaks features an Embedding Viewer powered by UMAP to provide an overview of all genomic regions and how they compare in terms of their similarity in the learned feature space. Finally, at the bottom right is the Progress View, which visualizes several properties of the interactively trained classifier to help users determine when to stop the labeling process. In the following, I'll show you snippets from a live recording where we trained a classifier from scratch to find asymmetrical peaks in a DNA-SNP H3K27 cell dataset that can indicate the directionality of an active promoter region. In order to build a classifier, we first need to specify a search query and then provide feedback to the system in form of binary labels. We start labeling regions selected by our active learning sampling strategy as either matching our search target, not matching our search target, or being inconclusive with clicks on the corresponding buttons. At the end of our initial sample set, we'll train our first classifier and then retrieve a new set of samples. As we continue in labeling regions and training classifiers, the progress view informs us about the state of our learned classifier. For example, we can see that the uncertainty gradually decreases. To gain an overview of all regions, we take a look at the embedding view. Using the prediction probability color map, we can quickly identify neighborhoods of regions that contain potential matches, showing here as these blue dots. We could now use the embedding view to explore the neighborhood in detail using lasso tool, but for now let's focus our attention to the results to assess the current performance of our trained classifier. Besides just browsing the results, we can further improve our classifier by continuing to label regions to confirm true positive predictions and to inform the classifier about false predictions. After several rounds of labeling and training, our labels can disagree with the classifier's prediction due to inconsistencies. Peaks notifies us about such conflicts to encourage resolving these issues. For example, here we fix some incorrectly assigned positive labels which we do not consider matches anymore. Once we are happy with the current results, we can explore the entire datasets in the query view. To guide this exploration, the query view visualizes the prediction probability as a light gray to blue heat map where dark blue represents strong matches. This way, we can quickly find other regions in the sequential data that exhibit similar patterns to our search query. To conclude, we showed that deep learning techniques can be used to augment human intelligence for smartly exploring epigenomic data. With peaks, it's relatively easy to train a classifier for finding and exploring epigenomic patterns that match the user's perceived notion of similarity. In general, our method should be regarded as a generic approach for pattern search that is complementary to specialized feature detectors to support more open-ended visual exploration. In the future, we want to explore other types of encoder models and compare the labeling efficiency of different active learning sampling strategies. For more details, please take a look at our paper and feel free to get in touch. Thank you.